Now that we have learned about the role of liver in the metabolism of carbohydrate, protein, vitamins, etc., now let's focus on something that is liver known to excrete. What I'm talking about is bile. First and most important thing to remember is that bile are synthesized in the liver and stored in the gallbladder. So what are some of the characteristics of bile? See here, bile is a bitter tasting dark green to yellowish brown fluid produced by the liver. Bile helps in the process of digestion of the lipids, especially in the small intestine. Bile is stored in the gallbladder, and upon the presence of food, it is discharged from the duodenum. Bile is mainly composed of water in major portion, bile salts and pigments. It also has fats, inorganic salts and cholesterol. Our liver cells secretes the bile in two stages basically. At first, the liver cells will secrete bile into canaliculi. From the canaliculi, the blood will move into the bile duct. And as our bile contains a large amount of bile acids, cholesterol, etc., they will flow from the bile duct, and as they flow, they will be modified and made watery. The gallbladder will store these biles and it will be concentrated 5 more times in the gallbladder by absorption of water. So what are the main contents then, and what are the main composition of the bile basically? So basically every day around 500 to 1000 milliliter of bile are secreted, and we have already discussed about their color. They can be transparent to light yellow in color. They have a pH of 7.8 to 8.6. Major contents are water. Then comes our bile salts. By bile salts, basically we are referring to mainly sodium and potassium of the bile acid. Third most important thing is the bile pigments. These are basically bilirubin and biliverdin. Other than that, it will contain fat, lecithin, fatty acid, cholesterol, etc. The bile salts, which is a major content of the bile, are also synthesized in the liver. Basically, they are synthesized from cholesterol, and there are two kinds of bile salts. Okay, we have our first one, the primary bile acids. These forms cholic acid and chenodeoxycholic acid. These bile acids go through the process of conjugation in the liver with taurine and choline, and as they get conjugated, cholic acid, if they get conjugated with taurine, they form taurocholic acid. If cholic acid gets conjugated with glycine, for example, they will form glycocholic acid. So the conjugation of tauro and glycocholic acid are mainly the reason that will lead to form sodium and potassium salt in bile. So sodium taurocholate and potassium taurocholate. These are known as the bile salts basically. The cholic acid and the chenodeoxycholic acid will be then in the colon. They will be acted by bacteria. And as the bacteria act on these in the colon, they will form something known as deoxycholic acid and lithocholic acid. These will be known as secondary bile acids. So you need to know the difference between primary bile acids and the secondary bile acids. The primary bile acids are those that are being synthesized in the liver from cholesterol, okay, with the help of the process of conjugation. The secondary bile acids are those which are actually taking place, which synthesis is actually taking place in the colon with the help of the bacteria found in the colon. Now, majority of these bile salts will enter the duodenum and will be absorbed by the portal vein and sent back to liver. This is known as enterohepatic circulation of bile salts. A minute amount, around 5 to 10%, will remain in the colon and again will be acted by bacteria to form deoxycholic acid and lithocholic acid. Again, these are known as secondary bile acids. In total, bile salts makes up 3.5 gram and they are continuously being recycled and sent back to liver. Usually in an ordinary meal, we need to have 6 to 8 gram of bile salts to digest and absorb fats. So during the process of digestion, the total bile salts will circulate twice to maintain their function of helping in digestion and absorption of fats. Then comes the third most major component of the bile called the bile pigment. These bile pigments are basically biliverdin and bilirubin. These are mainly will be formed by the destruction of our red blood cells. Okay, so from haemoglobin basically they will be formed when we are destroying the RBCs around that time. They will be excreted in bile and they are the one that is responsible for the yellow color of the bile. Now let's check out some important functions of the bile here. Number one, you should remember the features of bile. So bile acids are amphiphatic. They have both hydrophilic and hydrophobic properties. The cholesterol part of the bile acid will be hydrophobic, whereas the amino acid part will be hydrophilic. These features allow these bile acids to function or to perform two functions. The first one, it helps in emulsification of lipids. So basically in the dietary fat, the bile acid will act and will have something known as detergent action. This will cause the fat to break down into minute small droplets. And as they break down into small droplets, it allows the surface area of that particular GI area to increase. Okay, and as the surface area increases, it becomes more efficient. It helps the enzyme lipase to digest them at a very efficient manner. Okay, so this is one function that we have. The second function is solubilization and transport of lipid in an aqueous environment. Bile acids can carry lipids. So they can perform the function to make the lipids soluble by forming something known as micelles. What is micelles? These are basically aggregation of lipids that may include cholesterol, fatty acids, okay, and they will remain suspended in water. Bile also plays a role, very important role in the transport and absorption of our fat-soluble vitamins. The third function of the bile is in cholesterol metabolism. So the majority of the cholesterol in our body basically are broken down to synthesize bile. So in human, every day you can see here 500 milligram of cholesterol are converted into bile acids. This is a main or chief way of eliminating excess cholesterol from the body. In cases where we have inability to digest or ingest cholesterol. The last important function is, now we have known a good number of details about the bile. Let's now concentrate on our bile pigments, specifically on hemoglobin. In the last blood biochemistry chapter, we have learned in details about hemoglobin. So how is our bile pigment bilirubin and biliverdin basically will be formed? Let's check out this. So the process of catabolism of hemoglobin, that is breakdown of hemoglobin, will lead to the production of these pigments. How? So when the RBC have completed their lifespan, they will be mainly broken down. And as the RBCs are broken down, their hem group and the globin part of the pigment hemoglobin will be separated. The globin will be converted into several amino acids by breaking down. The methynyl bridge between ring 1 and ring 2 of the heme part will be broken down by heme oxygenase. And as it happens, at the methynyl bridge is broken down. If you remember, there was 4 pyrrole rings which were attached to one another by methylene bridge. That makes up a porphyrin, right? And hemoglobin was a porphyrin. So as methylene bridge is broken down by the heme oxygenase, it leads to release of iron. and converts the heme into the pigment called biliverdin, which is green in color. Now if you see this reaction, you'll have a better understanding. We have our hemoglobin here with iron at the center. The enzyme heme oxygenase will act on the methylene bridge, convert it into a pigment called biliverdin. The biliverdin that we have obtained now will be reduced by an enzyme called biliverdin reductase into bilirubin. So biliverdin gets converted into bilirubin. Bilirubin will be yellow in color, and this is the major pigment that is found in the bile, especially in humans. See following reaction, biliverdin gets reduced by biliverdin reductase into bilirubin. So if you hemoglobin basically will release globin, which will be converted into amino acid. Iron will be transported, and the biliverdin will ultimately be converted into bilirubin. Now, the bilirubin that is produced by this process are actually insoluble in water. And as they are not soluble and they are insoluble, they are known as unconjugated bilirubin. That is, they are not bound to anything. Now, in order to remove this bilirubin from the body, because it can be toxic, it can deposit in the tissues, we need to excrete them. So first we need to make them water soluble. To make them water soluble, we need to conjugate the bilirubin with something known as glucuronic acid. And conjugation with glucuronic acid will make them water soluble. Okay, and then ultimately it can be excreted from the body. Now, as the bilirubin gets conjugated with glucuronic acid, it will be no longer known as unconjugated bilirubin. Now it will be known as conjugated bilirubin. Now you can see here, after the hemoglobin is degraded and bilirubin is produced, we are calling it unconjugated bilirubin, which is insoluble. So what happens? They get attached with albumin and then are taken to the liver. In the liver, they get conjugated with gluconic acid and becomes conjugated bilirubin. This reaction is catalyzed by UDP glucuronyl transferase. Now from the liver, conjugated bilirubin will be excreted with bile, and then it will be entering the intestine. The conjugated bilirubin will be degraded by intestinal bacteria, which will convert the bilirubin into urobilinogen and stercobilinogen. Around 20% of the urobilinogen will be reabsorbed via the portal system to the liver, and then they can pass into general circulation or can be re-excreted into the bile. Now, if they get escaped into general circulation, they will be collected by kidney and ultimately excreted in the urine. And those urobilinogen which were not reabsorbed, the 80 other percent, they will be converted into stercobilinogen and ultimately will be excreted via fecal material. So see this diagram for a much more better understanding. RBCs getting destroyed, releasing heme, which ultimately is converted into biliverdin. Biliverdin then gets converted into bilirubin. The bilirubin that we are getting through this process is unconjugated and water insoluble. So what they do, they will bind with albumin and will be taken up by liver. In the liver, the unconjugated bilirubin will bind with glucuronic acid and becomes conjugated bilirubin. The conjugated bilirubin will be then excreted from the liver with bile and they will reach to the intestine. In the intestine, we have intestinal bacteria. They will convert the conjugated bilirubin into urobilinogen and stercobilin. Some of the urobilinogen will be reabsorbed and will be sent back to the liver via portal system, and some will be escaping in the blood. Those which will escape in the blood will be ultimately taken up by kidneys and excreted in urine. Okay, now this is all our bilirubin metabolism. Now let's see some clinical aspect of bilirubin metabolism. Jaundice, also known as hyperbilirubinemia, means excess amount of bilirubin. Hyperbilirubinemia, another name for jaundice. This is defined as yellow discoloration of skin, sclera, and mucous membrane. So basically, why does an individual may have jaundice? If increased bilirubin concentration, then the person may have jaundice. So normal range of bilirubin is around 2 to 3 milligram per 100 milliliter. But when it is above this level, we call it jaundice. Now, usually all our body fluids and body tissues are yellow in color, except for our brain. Because our brain has something called blood-brain barrier. This will not allow the bilirubin to go to the brain because bilirubin is toxic to the brain. So it won't allow bilirubin to have access. Except in cases of neonatal period. So in neonatal period, in children, they are at a risk if they don't have a control amount of bilirubin. If they have an excess amount of bilirubin, then these excess bilirubin can get deposited in. And if it happens, we call the condition Kernicterus. Okay, and it can cause several problems such as decrease of alertness, hypotonia, poor feeding, opisthotonus, the position as you can see here, this posture, and hypotonia. Now jaundice basically is of 3 kinds, and the main causes allows us to classify the jaundice into these 3 types. The first one is called hemolytic jaundice or prehepatic, also known as. Now the name suggests hemolytic, so they believe it happens because of excess breakdown of RBCs. Okay, the second type we have is called hepatic or hepatocellular jaundice. This is due to damage in the liver cells. The third kind we have is known as post-hepatic or cholestatic jaundice. This one is mainly due to obstruction of the bile duct. So basically the jaundice can be classified into these 3 types we have. So let's check out one by one of these 3 kinds. So the first one, hemolytic jaundice, also known as prehepatic jaundice. This is basically happening why? Because of excess production of RBCs. So if there is excess production of RBC, we have excess amount of unconjugated bilirubin. We don't have any problem with the liver. We don't have any problem with the bile secretion. Where is the problem? The RBC is being produced is excess, being destroyed is excess. So this is leading to an increased amount of unconjugated bilirubin. Now our liver cells cannot take up excess unconjugated bilirubin. It has also its limitation. So what will happen? The excess amount may remain in the blood. So this will lead to some unconjugated hyperbilirubinemia. It is taking, the liver is functioning properly, but it also has its limitation. So we are having the excess amount in the blood. So this is leading to unconjugated hyperbilirubinemia. Now this we know that is water insoluble, if you remember. Okay, and we knew that it has to bind with albumin to go to the liver. So since it is water, you won't find this bilirubin, this unconjugated bilirubin in your urine. So it won't appear in urine. You see here whether we'll have urebilinogen or not. See, as the liver is now working overtime because of excess production of destruction of RBCs and excess amount of unconjugated bilirubin, so the liver will also produce excess amount of conjugated bilirubin. So if we have excess amount of conjugated bilirubin, it means we'll have excess amount of urobilinogen. So in our urine, the urobilinogen amount will increase. Similarly, if you see fecal stercobilinogen, usually the normal range is 25 to 250 milligram per day. But now, as we are having more conjugated bilirubin, the liver is producing, we'll have more stercobilinogen. So patients who will have this kind of jaundice, hemolytic jaundice, due to the stercobilinogen amount being increased, their feces will now become dark brown in color. Okay. So what is happening? Number one, there's increase of unconjugated bilirubin. Number two, there won't be any bilirubin in the urine because the unconjugated bilirubin is water insoluble. However, there will be urobilinogen increased in the urine, stercobilinogen increased in the urine. Amount of fat, fecal fat level will be normal. The second kind we have is the hepatocellular jaundice. And why it's happening? Mainly the liver have some problems, okay, maybe due to liver diseases or liver damages, so they won't be able to conjugate and transport the bilirubin into the bile duct. So mainly there is something problem with liver. Okay, there is no problem with RBCs being excessively destroyed and so on. The main problem is here with the liver. As a result, the liver cannot uptake the unconjugated bilirubin and also cannot make conjugated bilirubin. Cannot pass or excrete the conjugated bilirubin. So there will be both conjugated and unconjugated bilirubin will increase in the blood. And we know that conjugated bilirubin is water soluble. Okay, as a result, find bilirubin in the urine. Okay, and such cases is also known as choleuric jaundice as well. Now, urobilinogen decreased. Why? Because the damaged liver cells are producing and excreting less conjugated bilirubin than normal. So we'll have a decrease urobilinogen, we'll have a decrease stercobilinogen. So feces will become pale color. Previous one was dark, now this is becoming pale color. The fat in the feces will increase. Why? Because less bile is being sent to intestine, so less emulsification and absorption of the fat is taking place, so more fat will pass through the feces and so there will be excess amount of fat. Okay, this excess amount of fat that is being discharged through the feces is known as steatorrhea, discharge of fat via the feces. So the feces will be bulky, pale, greasy and have foul smell. One we have is the cholestatic or obstructive jaundice. Now this is mainly due to if there is any obstruction in the bile. Okay, so if there is any obstruction in the bile duct, so bile cannot pass through. So in these cases, the liver has no problem. Liver is uptaking normal amount of unconjugated bilirubin. Liver is producing normal amount of conjugated bilirubin, but the bilirubin conjugated cannot pass because the bile duct is obstructed. And we only have conjugated hyperbilirubinemia, mainly due to impairment of flow of the bile. So if we have conjugated hyperbilirubinemia, okay, and if there is less conjugated bilirubin is being passed to intestine, we'll have a very decrease of urobilinogen and stercobilinogen in the urine. However, as conjugated bilirubin is being produced but at a small amount, still we'll find the urine in the bilirubin in the urine. But decreased level of urobilinogen, okay, have decreased level of stercobilinogen, totally absent. So the stools will be clay color. The amount of fat will be also greatly increased in the stool. Okay, so these are our three kinds of jaundice: hemolytic, hepatocellular, and post-hepatic or obstructive. All are all listed in this table. It's very important table. Must know this table. High yield. Okay, so please do remember here. Okay, so for this chapter, This is one of the important yield products. Okay, keep in mind one more thing, that the conjugated bilirubin that we know, this also has another name called direct bilirubin. The unconjugated one that we have is also known as indirect bilirubin. So with this, we have finished our liver biochemistry. The important things to know is the role of liver in metabolism, that is of lipids, proteins, glucose, and so on. Their role in detoxification, so please know about phase 1 and phase 2, very very important. High yield, okay, do remember about phase 1 and phase 2. Know about the characteristic features of the bile and their functions. What is primary bile acid and secondary bile acid, you must know. What are the bile pigments, that is biliverdin and bilirubin. The function of the biles you must know. Okay, and lastly, the catabolism of haemoglobin and the jaundices that we have learned today, the 3 kinds. Okay, so please pay your focus on these contents here. So that's all for today. Thank you.